And the question is, the classification system proposed by Linnaeus was a dash kingdom system of classification. 2, 3, 4 or 5. And the answer for this is 5. Let's know how. Before 1969, all the organisms had been divided into two groups, that is plants and animals. Now, mainly on the basis of presence or absence of cell wall, Linnaeus, who is known as father of taxonomy, founded two kingdoms, Kingdom Plantae and Animalia. Animalia in his two kingdom system of classification. Let's move on to the next question. In Whitaker's five kingdom system of classification, eukaryotes are distributed among two kingdoms, three kingdoms, four kingdoms, or all the five kingdoms. And the answer for this is four kingdoms. In Whitaker's five kingdom system of classification, eukaryotes are distributed among four kingdoms. Now let's know which are those four kingdoms. Protista, Plantae, Fungi, and Animalia. These are the four kingdoms which was distributed by karyotes in Whitaker's five kingdom system of classification. Four kingdoms: Protista, Plantae, Fungi, and Animalia. Let's move on to the next question. Dash is the most common method of reproduction in bacteria. Binary fission, endospore formation, conjugation, or sexual reproduction. And the answer for this is binary fission. So reproduction in bacteria occurs by three methods, that is binary fission, sporulation, and sexual reproduction. Now you may also get the question as dash are the most common methods of reproduction in bacteria. They may give you these three options with the option fourth, that is all of these. So you have to take all of these. Now right now for this question, dash is the most common method of reproduction in bacteria is binary fission. And then binary fission is the most common method of bacterial multiplication under favorable conditions. Bacterial multiplication under favorable conditions in which a mature bacterium divides into two equal daughter cells. So binary fission is the most common method of reproduction in bacteria. And what is binary fission? Binary fission is a bacterial multiplication under favorable conditions in which a mature bacterium divides into two equal daughter cells. So bacterium is single, bacteria is two or more. Let's move on to the next question. Cyanobacteria are classified under which of the following kingdoms? Monera, Protista, Algae, or Plantae? And the answer for this is Monera. Let's know how. Cyanobacteria or blue green algae are gram positive photosynthetic prokaryotes which perform oxygenic photosynthesis. Photosynthetic pigments include chlorophyll A. Let's write it down. Chlorophyll A, carotenoids, and phycobilins. Cyanobacteria are classified under kingdom Monera. Cyanobacterial cell structure is under kingdom Monera. Cyanobacterial cell structure is typically prokaryotic. One envelope organization with peptidoglycan, CAN, wall, naked DNA, 70S ribosomes, and the last but not the least, absence of membrane bound structures like endoplasmic structure, mitochondria structure or reticulum. Mostly write endoplasmic reticulum and not structure. And then comes mitochondria. It's not mitochondria, it's mitochondria, Golgi bodies, and many others. There are plastids, there are lysosomes, there are sap vacuoles, and the other or the outer part of the protoplast called the chromoplasm contains a number of photosynthetic thylakoids. And the next question is mycoplasmas are classified under which of the following kingdoms? Monera, Protista, fungi, or Plantae? And the answer for this is again Monera. Let's know how. Mycoplasma or molecules are the simplest and the smallest of the free living prokaryotes classified under cattle suffering from pleuropneumonia, knockout or rooks. Knockout or rooks. Now, they mostly produce pneumonia in domestic animals. A typical pneumonia or pneumonia and mycoplasmal urethritis in humans, little leaf disease of brinjal and which broom implants mycoplasm can survive without oxygen. And the next question is dash are important decomposers that cause decay and decomposition of dead bodies of plants and animals. The options are as follows: saprophytic bacteria, saprophytic fungi, plants like Sarracenia, or both saprophytic bacteria and saprophytic fungi. And the answer for this is both A and B. Let's know how. Saprophytic bacteria, free living bacteria which obtains their food from organic remains. For example, corpses, excreta, fallen leaves, vegetables, fruits, meat, jams, etc., and even bread and other products of plant and animal. And then breakdown of organic compounds is called as decay. Nature saprophytic and bacteria along with saprophyte trophic are the decomposers of organic remains. So therefore Option A and option B, that is saprophytic bacteria and fungi, are the important decomposers that cause decay and decomposition of dead bodies. And the next question is photosynthetic pigments of cyanobacteria, which is also called as blue-green algae, includes chlorophyll A, carotene, xanthophylls, or all of these. And the answer for this is all of these. We had seen in the last question that some question chlorophyll A, carotenoids, and xanthophylls. Cyanobacteria or blue-green algae are gram-positive photosynthetic prokaryotes which performs oxygenic photosynthesis. Now, what does photosynthetic pigment contain? This is very important. You may also get the question of photosynthetic pigments of bacteria, which may include pigments are as follows: chlorophyll A, carotenoids, and xanthophylls. Cyanobacterial cell structure is typically prokaryotic, one envelope organization with peptidoglycan wall, naked DNA, 70S ribosomes, and absence of membrane bound structures like endoplasmic reticulum, mitochondria, Golgi bodies, plastids, lysosomes, sap vacuoles. The other part of the protoplast, called chromoplasm, contains a number of photosynthetic thylakoids. Let's move on to the next question. Cyanobacteria are used in agricultural feeds for crop improvement because they cause endofixation, algal blossoms, photosynthesis, or all of these. And the answer for this is all of these. Let's know how. Heterocyst is a large-sized, pale-colored, thick, several cyanobacteria have the ability of nitrogenous fixation. The filamentous forms possesses a special large cells of heterocysts for this. Some of the fixed comes out as excretion. After death of cyanobacterial substratum becomes rich in nitrogen, such nitrogen fixation cyanobacteria are now regularly inoculated in the rice fields. This saves the application of nitrogen fertilizers. So the answer for this is nitrogen fixation, algal blossoms, and photosynthesis, that is all of these. Cyanobacteria are used in agricultural feeds for crop improvement because they cause what? Means all of these. Let's move on to the next question. The 5-kingdom classification system, and the kingdom that includes the blue-green algae, which is also called as, yes, cyanobacteria, nitrogen-fixing bacteria, and methanogenic archaebacteria, is plantae, fungi, protista, or monera. And the answer for this is monera. Guys, monera kingdom is the most important in this whole chapter, so learn it properly. Yeah, let's come back to the question. Yes, so monera kingdom includes prokaryotes, mycoplasm bacteria, actinomycetes, and cyanobacteria, or blue grain algae. Along with fungi, they are decomposers and mineralizers of the biosphere. Let's note it down here. Along with fungi, they are decomposers and mineralizers of the, it actually looks like strepsils, but this is biosphere. Let's move on to the next question. The kingdom Protista forms a link with kingdom Plantae, fungi, animalia, or all of these. Now, what do you think? Protista, Plantae, fungi, animalia. In fact, all of these. It forms a link with kingdom Plantae, kingdom fungi, and kingdom animalia. And the next question is, which of the following kingdoms has no well defined boundaries? Monera, Protista, fungi, or Metaphyta and Metazoa? And the answer for this is Protista. Let's know how. Although all single-celled eukaryotes except some unicellular algae, example, Chlamydomonas, chlorella, or fungi. Eastern animals which are placed in their respective kingdoms along with their closely related multicellular organisms are placed in kingdom Protista, which has yet its boundaries are not well defined. Let's move on to the next question. Members of kingdom Protista are primarily parasites, terrestrial, fungi, or photosynthetic. And the answer for this is, what do you think? It's fungi. This looks like mushroom. Yes. So, protists are mostly aquatic organisms, therefore, they are found in the sea or the fresh water, and that is moist soil. Many protists are found in water bodies in the form of plankton. Some protists live in the bodies of animals as parasites. And let's move on to the next question. Which of the following organisms have been placed under kingdom Protista? Option number A, chrysophytes and dinoflagellates, euglenoids, slime molds and protozoans, or all of these. And the answer for this is all of these. Let's know how. Listen very carefully to the explanation of this question. The kingdom Protista has been broadly divided into three main groups. That is photosynthetic protists, example dinoflagellates, chrysophytes and euglenoids. Number 2, consumer, that is decomposer protists, which are also called as slime molds. Option number 3, the last one, that is protozoan protists. Now let's know what are euglenoids. Now these are euglena. These are euglena, like single unicellular flagellates which possesses pellicle instead of cell wall. Chrysophytes include diatoms or desmites. Chrysophytes includes diatoms or desmites. Now, they belong to the division Chrysophyta or Bacillariophyta. The dinoflagellates belong to division Phyrophyta and class Dinophyceae. Slime molds possesses the characters of both animals and fungi, and therefore they are commonly called fungus animals. Okay, which of the following groups of organisms are included under chrysophytes? As we saw in the last question, that is chrysophytes, diatoms and desmites. So, which of the following groups of organisms are included under chrysophytes? Diatoms and desmites, which is also called as golden algae. Diatoms of dinoflagellates? No. Alganoids? No. Or slime molds? So the answer for this is yes, diatoms and desmites, which is also called as golden algae. You may also get the question as this. And let's move on to the next question. The cell wall is composed of two thin overlapping shells which fit together like a soap case. Okay, it looks like a mitochondria in a box. Okay, forget it. So that's soap, okay? The cell wall is composed of two thin overlapping shells which fit together like a soap case. Which are they? Desmites or diatoms, dinoflagellates or slime molds? And the answer for this is diatoms. Let's know how. The body of diatoms is covered by transparent siliceous shell. Siliceous shell. मतलब if this is the outer cover, this is the shell, which shell? Siliceous shell, and known as frustule. This is also called as frustule. This is very important. Also called as frustule. Now let's know what is frustule. Frustule is made of two valves. Two valves. Which is epitheca or hypotheca. The two valves fit together like a soap box. The frustule possesses very fine markings, pits and pores. So the cell wall is composed of two thin overlapping shells which fit together like a soap case in desmites, diatoms or dinoflagellates or slime molds. And the answer for this is yes. How? Now, what is siliceous shell? It is a cover of a soap. Okay. It's a cover of the soap. It is just like the outer part of the soap case. Now, what is siliceous shell? It is also called as fistule. Now, fistule is made up of two parts, that is epitheca and hypotheca. Let's move on to the next question. Diatomaceous earth is used for all except polishing, filtration of oils and syrups, making sound in fine-proof rooms, or biogas products. Okay. Polishing, filtration of oils and syrups, making sound in fine-proof rooms, or biogas products. And the answer for this is biogas production. Let's know how. Diatomite deposits are often accompanied by petroleum fields. Much of the petroleum of today is probably due to decayed bodies of the past diatoms. Diatomite is porous or chemically inert. It is therefore used in filtration of sugar, oils, alcohols, syrups, or antibiotics. Diatomaceous earth is used to make soundproof rooms. Diatomite is employed as a cleaning agent in toothpastes and metal polishes. Chrysophytes are planktons, nectons, lenthetic organisms, or rooted submerged. And the answer for this is planktons. Let's know how. Diatoms are very important photosynthesizers. About of all the half organic matter synthesized in the world is believed to be produced by them. Diatoms are probably the most numerous of all plants like protists. Because of this abundance, they are one of the most important primary producers of the seed. There are about 5,500 species of diatoms. Mainly marine, the diatoms constitute an important phytoplankton component of the ocean. And the next question is siliceous fistules of diatoms being indestructible piled by up at the bottom of ocean and formed a thick bead or bed. Over billions of years, such a thick bed is known as most important question. Diatomous earth. Let's know how. The diatom shells piling up at the bottom of oceans. Okay, bottom of oceans. Now this is the ocean, this is the surface of the ocean, and this is the middle part, and this is the bottom. Okay, bottom of oceans over millions of years and years and years form deposits called diatomaceous earth. Now these deposits may be 1,000 meters thick with as many as 5 million diatom shells in each cubic centimeter. This diatomaceous earth is used in many small and large industries. Let's move on to the next question. Dinoflagellates are mostly marine and saprophytic, freshwater and photosynthetic, marine and photosynthetic, or terrestrial and photosynthetic. And the answer for this is marine and photosynthetic. Let's know how. Dinoflagellates are a group of 1000 species of photosynthetic protists. Dinoflagellates are a group of about 1000 species. 1K is 1000. Yeah. So 1000 species, 1000 species of the dinoflagellates are important components of phytoplankton. The dinoflagellates are important components of phytoplankton. Most of them are marine, but some occur in fresh water. Some dinoflagellates, such as, now these are most important, that is gymnodium and gonialox. Now these two species grow in large number in the sea and make the water look red and cause the so-called red tide. So why do you call Red Sea Red Sea? Because these two species, that is Gymnodium and Gonialox, are present in the sea. That's why called as Red Sea. No one has added red color to the sea. These two species are present. So that's why we call as red tide or Red Sea. Let's move on to the next question. Red tides in warm coastal water develop due to the abundance of dinoflagellates, euglenoids, diatoms and desmites, or slime molds. And the answer for this is dinoflagellates. Now let's know what is dinoflagellates. Dinoflagellates are a group of about 1000 species of photosynthetic protists. So, the dinoflagellates are important components of phytoplankton. Most of them are marine, but some occur in fresh water. So, there are two species, that is Gymnodium and Gonialox, grow in large number in the sea and make the water look red and cause a so-called red tide. Let's move to the next question. Multinucleate slimy mass of protoplasm which forms the body of slime molds is called as Plasmodium, Maximoeba, Phorocytes, or Periplasmodium. And the answer for this is Plasmodium. So euglenoids are unicellular flagellate protists commonly found in freshwater ponds, pools, and moist mud. They lack cellulose cell wall. The body is covered by thin and flexible pellicle. What is pellicle? Periplast. So the pellicle has oblique but parallel strips, which are called as myonemes. The euglenoids have two flagella, usually one long and one short. So, though the euglenoids are photosynthetic in the presence of light, when deprived of sunlight, they behave like heterotrophs by predating on smaller organisms. Holozoic or feeding is called mixotrophic. That is a mix of holophytic and saprobic or holozoic. Photosynthetic pigments of euglenoids are identical to those present in higher plants. For example, chlorophyll A and chlorophyll B. Let's move on to the next question. Slime molds are photosynthetic protists, saprophytic protists, or both A and B, or none of these. And the answer for this is saprophytic protists. Let's know how. Slime molds do not have chlorophyll. They are surrounded by the plasma membrane. Only somatic parts are surrounded by the plasma membrane. Only somatic parts are without cell walls. The slime molds live usually amongst decaying vegetation. They are quite common on lawns and moist fields. They exhibit wide range of coloration. They are phagotrophic or saprotrophic nutrition. They have phagotrophic or saprophytic nutrition. Parasitic forms are not known, but asexual and sexual modes of reproduction are found. They produce spores. This produces spores within sporangia. Sporangia. A spore possesses a cell wall called cellulose. And you've got the answer plus the explanation with this. Let's move to the next question. Causal organisms of sleeping sickness and Kala Azar belong to which of the following groups of protozoan protists? Amoeboid protozoans, flagellated protozoans, ciliated protozoans, or sporozoans? And the answer for this is flagellated protozoans. Let's know how. Trypanosoma gambinis causes gambian sleeping sickness. The parasite is transmitted by blood-sucking tsetse fly. Glossina palpalis causes kala azar or dum-dum fever, which is also called as visceral leishmaniasis. The parasite is transmitted by sandfly, that is Phlebotomus argentipes or other species. Causal organisms of both diseases belong to flagellated protozoans of kingdom Protista. Let's move on to the next question. The sporozoa are all internal dash that typically have an infective cyst stage in their life cycle. An example of sporozoa is the genus dash, which causes malaria. The options are ciliates plasmodium, flagellates plasmodium, parasites plasmodium, or parasites trypanosoma. And the answer for this is parasites plasmodium. How did we get this answer? Let's check it out. All sporozoans are endoparasites. Sexual reproduction takes place through syngamy. Asexual reproduction occurs through multiple features. Under unfavorable conditions, they form cysts. For example, plasmodium, which is already given, monocystis. Let's move on to the next question. Which out of the following is a ciliated protozoan? And the options are Plasmodium vivax, Amoeba proteus, Paramecium cordatum, or Leishmania donovani. And the answer for this is Paramecium cordatum. Let's know how. Paramecium is a free-living ciliate which is found in fresh water. Most widely distributed species Paramecium cordatum and Paramecium aurelia. These two are the distributed species. The cilia of the extreme posterior end are longer and form bunch called caudal tuft. Called caudal tuft. Let's move on to the next question. Dash is a parasite of large intestine of human beings and causes the disease dash. Escherichia cell, amoebic dysentery, entamoeba histolytica, amoebic dysentery, Plasmodium vivax malaria, or Trinosoma gallens sleeping sickness. And the answer for this is entamoeba histolytica. Now, entamoeba histolytica resides in the upper part of the human large intestine and causes the disease amongst dysentery or amoebiasis. The symptoms of the disease: abdominal pain, repeated motions with blood and mucus. It feeds on red blood cells, that is RBCs, by damaging the large intestine and reaching the blood capillaries produces ulcers in the body as well as mostly large intestine. Let's move on to the next question. Fungi shows asexual reproduction by all of the following kind of spores except Conidia, Eospores, Sporangiospore or Zoospores. And the answer for this is oospores. So, asexual reproduction in fungi occurs through formation of spores. Spores are single-celled protoplasts which separates from the parent organism and causes dispersed. They may be motile or non-motile and thin-walled or thick-walled. Asexually produced, resemblance the parent body in having some sets of chromosomes or genosomes. These are produced after meiosis in some fungi, which are as follows: ascospores, basidiospores, oospore. These are the product of sexual reproduction. Let's move to the next question. Dikaryophase is a specific characteristic of all fungi, Phycomycetes and Ascomycetes, Basidio and Deuteromycetes, Ascomycetes and Basidiomycetes. And the answer for this is Ascomycetes and Basidiomycetes. Now, in Ascomycetes and Basidiomycetes, there are an intervening phase called dikaryophase between plasmogamy and chirogamy. Intervening phase. So the phase, this is the phase, okay? And this is called as dikaryophase. Now in this phase, the cells contain two nuclei or dikaryons, N plus N. Such cells are called dikaryotic cells. In such fungi, the life cycle is complete and three phases instead of two haplophase, dikaryophase and diplophase, Zn or 2n halophase, by plasmogamy converts into dikaryophase where each cell has two haploid nuclei. Then chirogamy occurs, converting dikaryophase to diplophase. Meiosis occurs in diplophase and haploid condition is regained. Let's move on to the next question. Eukaryotic achlophysis and heterotrophic organisms. Monera, protista, fungi or plantae? And the answer for this is fungi. Let's know how. Fungi is a large kingdom of over 1 lakh species. They are achlorophyllous, heterotrophic, spore-forming, non-vascular and eukaryotic organisms which often contain chitin or fungal cellulose in their walls and possesses glycogen as food reserve. They are cosmopolitan in occurrence, being present in air, water or soil, over and inside animals and plants. Cenocytic mycelium is uninucleate septate, multinucleate septate, multinucleate aseptate, or both B and C. And the answer for this is multinucleate aseptate. So the cenocytic mycelium is multinucleate aseptate. Multinucleate is many cells. Aseptate is what? Absence. So absence of septate. Let's move on to the next question. Fungi lacking cross walls in the mycelium belongs to the class Phycomycetes, Ascomycetes, Basidiomycetes or Deuteromycetes. And the answer for this is Phycomycetes. Let's know how. Phycomycetes, lower or algal fungi, are characterized by aseptate cenozytic hyphae. Asexual reproduction by zoospores or aplanospores produced endogenic inside progenia. Sexual reproduction is isogamic and heterogamous. Heterogamous reproduction is of two anisogamous and oogamous. Phycophyta or Phycomyta is divisible into two groups, that is oomycetes and zygomycetes. The mycelium is cenocytic, that is multinucleate and aseptate. Let's move on to the next question. Which one of the following is an incorrectly matched pair? Phycomycetes, Mucor albugo, Ascomycetes, Penicillium aspergillus, Basidiomycetes, Pucania and Agaricus, or Deuteromycetes, that is Eustalegia or Conetrotrichum. And the answer for this is Deuteromycetes, that is Eustalegia and Coletrichum. Let's know what is Deuteromycetes. It is an artificial class of fungi which has asexual reproduction often occurs by conidia along with some other types of spores. In some cases, even asexual spores are absent. Eustalago is the member of Basidiomycetes. Let's move on to the next question. Term used for this closed ascocarp is apothecium, ambithecium, endothecium or clipstothecium. And the answer for this is cliptothecium. Let's know how. Ascus is a sporangial sac peculiar to Ascomycetes. 4 to 8 haploid ascospores are produced internally in each ascus. The asci may occur freely or get aggregated with dikaryotic mycelium to form fructifications called ascocarps. Ascocarps are of many types. Cup-like, which is also called as aptothecium, for example, Pezisa, flask-shaped perithecium, for example, Neurospora, elongated with a slit, Historia thecium or closed clistothecium, for example, penicillium. Let's move to the next question. Main component of the cell wall of fungi is cellulose, pectin, chitin or dextrin. This is the most important question and the answer is chitin. Let's know how. In fungi, cell wall contains chitin or fungal cellulose along with other polysaccharides, proteins, lipids and number of other substances. Chitin is basically made up of N-acetyl glucosamine. In some fungi, cellulose is also present either alone, for example, Phytophthora and many other oomycetes, or along with chitin. Let's move to the next question. Virion is nucleic acid of virus, antiviral agent, protein of virus or completely assembled virus outside host. And the answer for this is virion is protein of virus. Let's move on to the next question. Which of these is a defining character of plants? Autotrophic nature, eukaryotic cell of structure, cellulosic cell wall or aerobic respiration? And the answer for this is D, that is aerobic respiration. Let's know how. Virus is a non-cellular infectious nucleoprotein entity enclosed within a protein covering or coat. It is able to utilize the synthetic machinery of a living coat. It is able to utilize the synthetic machinery of a living cell of another organism for its multiplication, which does not involve growth or division. They have either RNA or DNA as genetic material, but never both. Virus is an obligate parasite and is inert outside the host. An inert virus outside host is called virion. It can be crystallized and stored. The smallest self-replicating infectious RNA parasites, they are known to cause diseases in plants only. For example, potato or spindle tuber disease. So aerobic respiration is a of plants. Let's move on to the next question. Which of the following is not a viral disease of plants? Red rot of sugarcane, tobacco mosaic disease, leaf curl of tomato, or Triticza disease of citrus. The answer for this is Let's know how. Fungus, that is the fungus, this is the fungus which is there in red root of sugarcane. Now this fungus produces red root sugarcane, which is conspicuous on leaf, midribs, or as well as in the canes. It reduces the juice content of canes and brings about withering of leaves. So, the red rot of sugarcane is not a viral disease of plants. Tobacco mosaic virus is a viral disease. Leaf curl of tomato is also a viral disease of plants. And tristeza disease of citrus is also viral disease. This is a fungus. This is a fungi virus disease. So, Clostridium falcatum fungus is in the red rot of sugarcane, which reduces the juice content of canes and brings about withering of leaves. Who crystallized and isolated viruses for the first time? W.M. Stanley crystallized and isolated viruses for the first time. Viral diseases can be classified as follows. Plant viral diseases, that is potato mosaic, pumpkin mosaic, apple mosaic, tulip mosaic, or bhindi mosaic, tulip, bhindi yellow bean mosaic, potato leaf roll, papaya leaf curl, banana bunchy top, or tomato bunchy rice tungro, etc. Animal viral diseases, that is foot and mouth, rinderpest, ranikhet, or Newcastle or fowl bird flu, etc. Human viral diseases, measles, smallpox, mumps, poliomyelitis, rabies, hepatitis, dengue, encephalitis, AIDS, common cold flu, herpes, SARS, that is severe acute respiratory syndrome, bird flu, H5N1, swine flu, H1N1, etc. Now, Dmitry Ivanovsky discovered that the cause of tobacco mosaic disease could pass through a porcelain filter. Temin and Baltimore discovered the retrovirus and it could pass through the porcelain filter, and the phenomenon of Teminism is 1972. Prions are highly resistant glycoprotein particles which function as infectious agents. They are also formed due to mutation in gene PRNP. Prions can also act as catalyst converting normal protein into prion state. Prions are not affected by proteases, nucleases, temperature up to 8000 degrees Celsius, UV radiations, and formaldehyde. Prion was discovered by Prusiner in 1983 for the first time. Virus is a non-cellular infectious nucleoprotein entity enclosed within a protein covering. A biosynthetic machinery is absent of virions are the smallest self-replicating infectious RNA particles which are devoid of protein coat. They are obligate parasites. They are known to cause diseases in plants. For example, potato or spindle tuber.